test is on Tuesday after break, right?
Yep.
There's a study guide online now. Just put it. If it's not there, let me know, because I think it's there. So, let me know if you have any questions, that kind of stuff. I will say that I probably won't respond to anything until about Wednesday, because I won't be in town. So, but after that, that email is the best. And I might be around here a little bit, but you probably don't want to come here to find me on your break. So who knows where you'll be. But wherever you are, take some time to rest, relax, break. So the test will include, let's see, we sort of started just after the neutral theory and then testing for selection. I think we started off, codon bias, all that kind of stuff, all the way up to what we talked about today, which will be sort of some aspects of human health and evolution. There's a sort of interesting book, I guess, called Survival of the Sickest. It's out there if you're sort of interested in this kind of topic. There's also all kinds of other things, but that's one I read a few years ago. It's not the greatest, but it's not bad either. I think the person who wrote it sort of sometimes goes a little beyond what the data really says, but I guess you're allowed to do that. Anyway. But it's about the evolution of disease and how some diseases are more prevalent in certain populations and why. Because, as you know, if something sticks around, maybe it has some advantages, or maybe some aspect of it has some advantages. So he sort of delves into that. So survival of the sickest for more information. But let's start with pathogens, those sort of critters that are going to cause harm. The pathogens are organisms that cause harm to the host, right? Forgot the right one. Harm to the host. All right, now, many of these pathogens live in large population sizes and have very short generation times, especially considering the generation time of the host, and have high mutation rates, which also includes a way of sort of getting recombinant DNA. Many bacteria and viruses, virus pathogens, will sort of pick up DNA from the environment. If you've had microbiology, you're aware of this, or maybe some other classes, but there's something out there called horizontal gene transfer. So normally, you sort of learn about DNA gets passed from one generation to the next, and blah, blah, blah. Right? That would be vertical transmission of DNA. Horizontal transmission or horizontal gene transfer is referring to sort of movement of DNA sort of within the same generation, not, so, and bacteria are renowned for just sort of picking up DNA and plasmids, things like that. They also have sort of conjugation mechanisms for exchanging DNA as well, but horizontal gene transfer is a sort of real big player, as it were, in bacterial populations and the bacterial genomes. Even more so than we used to recognize it. It was sort of like, yeah, this is probably important. Now they've decided it's really important. So they tend to get a lot of variation, and so pathogens evolve quickly is what that sort of means, relative to their hosts, generally speaking. And why do they need to evolve? What is the host doing?
Immune response.
Mounting immune defenses. So one class that you might sort of, I've always found interesting, although as far as vocabulary goes, it's an extremely difficult class, and that's immunology. It's sort of like War of the Worlds type thing going on. It's very interesting stuff. The hardest part of the class, though, is getting the vocabulary straight. But it is a very interesting class. That and virology. Again, a lot of jargon, and if you can cut your way through that, I think you can find it very interesting. Now, so the flu virus. You have to, how many people got their flu shot? So we're not going to make it a political discussion, but yeah, a lot of people get flu shots. So there is definitely some arguments for and against or whatever, but generally speaking, the CDC and all say, why not? Why don't you just go ahead, right? But why do you got to get one every year? Because other vaccinations, you don't need to do that.
Is it mutated?
No, it's a pharmaceutical
Oh,
stamp.
right. They have a patent on it, though, for that year.
No, right?
They have a patent on it for that strain. Usually.
Well, what's the flu doing? Why does the vaccine work forever?
Because it's a
It's different,
Yeah, this flu, yeah.
the flu.
But they changed, right?
Right.
Flu viruses changed. So you might be sort of creating antibodies for one thing, but the flu you see has got sort of a different mask on, and your immune system goes, you look familiar, but go ahead. So the flu is always changing. But the question really becomes this. Is the flu adapting, or is it simply evolving by neutral means?
Neutral means they're really bad at copying their genome, so they mutate simple.
How would you know? You have the tools in your toolbox, you know, believe it or not, to sort of design a sort of quick study, or at least understand the kinds of information you would need to know to sort out, is the flu, as it sort of changes every year, is it evolving by adaptation or natural selection, which is leading to adaptation, right? That's the mechanism that leads to adaptation. Or is this sort of a random drift neutral model? Is it really evolving by that? How would you know?
I think you could argue that, you know, the rapid mutation rate could be an adaptation. I mean, it's
Well, that's definitely going to provide plenty of variation if it is selection.
right.
But
Well, isn't it both? Like, wouldn't you have, it would adapt because, I just,
It's gonna evolve just by
because
doing good anyway. It's gonna evolve regardless. Because it's, the autoimmune defenses may evolve more rapidly. There's like two, I guess, different.
Well, evolving, sure, but
the
adapting?
recombinant, it would have the recombinant, and like that would make it random, I suppose. But it would adapt as well.
once
Well, we are, so
our definite
we've already established a while ago that, remember, the generation of variation is just normal, right? But now I'm asking you about the fate of that variation, which is it evolving by drift, or is it evolving by natural selection? Or is the flu virus, every year that we see it, has it evolved from the last time we saw it by natural selection, or by random drift?
I would say random drift.
One vote for random drift. You can sort this out. You have the power, you have the tools, you have the know-how. Think about what you know. What information would you need to sort that question out?
Define natural selection.
Does drift really play such a big role in it?
Well, good point. High population sizes, drift has little effect.
Are non-living particles affected by natural selection?
Non-living particles? Oh, now let's not get into viruses living or dead. They definitely replicated, whether they're living or dead, hijack your DNA and make it do things for it.
Right.
So definitely, so let's just call it sort of pseudo-living.
Like test selection, right? We would have to test selection.
Ah, you could test for selection, couldn't you?
Right.
I'd want to know its fitness.
Well, that would get to after testing for selection, right? Because in order to have selection, you need differences in fitness among some genotypes. But what test of selection are you referring to?
I was just touching on it.
Okay, I'll give you a hint. Big Macs.
Big one.
The restaurant.
Such map.
McDonald, Kreitman.
Oh.
Didn't we cover the McDonald Kreitman test?
Kind of touched on it.
At least we probably covered this idea.
Yeah.
Non-synonymous. That's
Okay.
what you're getting at, right? Right? We can at least begin to look for selection at traits and look for differences in fitness, but get down to the molecular level and test has selection been acting on some stretch of DNA or not? Yes? Do you remember how this plays out? This is the non-synonymous ratio of non-synonymous and synonymous. And if it's less than 1, greater than 1, so we'll just sort of dichotomize it for now. And also, the McDonald-Kreitman test is a modification of this. We did cover that, didn't we? right, where you do like polymorphisms and that kind of stuff. Just sort of, but just for now. So that's one place to start. Yeah? You could say, now what part of the genome? Viruses don't have huge genomes, right? What, did you have to learn about that anywhere? Did anybody learn about that? What do they have in their genome?
I did.
Like a retrovirus might have a reverse transcriptase, of course.
And RNA for its genome.
Well, some viruses are DNA, some are
Right.
RNA.
Well,
But what do they have around them?
Capsules.
Yeah, the protein code of the capsule, the protein capsule. So they have a gene that's going to sort of make that, a few genes that are going to make that, right?
matrix,
What else they got?
Segment.
The most important part,
Receptors, attachment receptors.
Cells,
the receptor. Huh?
the part that goes in the cells, I just.
The part that goes in the cell, that's the DNA or RNA, depending. So, but you know, there's a mechanism to inject it in there. But how does it latch onto the cell?
Receptors.
Receptors, right? The antigenic sites. Do you know that word? Antigenic sites. The antigenic sites are the sites that are going to interact with the host cells, right? So if you sort of imagine a virus and it's got the receptors sticking out, just like, you know, any classic cell membrane's always got stuff sticking out of it, right? So the receptors are the antigenic sites. These are the part that the immune system is going to recognize because that's what the antibody is made to stick to, right? When your body or the immune system makes antibodies, it's targeting the antigenic sites of the pathogen, correct? That in biology hasn't changed as far as I know yet. Things change. I might say something. So if we take this sort of idea and look at antigenic sites and do this McDonald-Kreitman test or this sort of, this DN-DS ratio, you can begin to sort out. Have the antigenic sites actually been evolving by adaptive selection, or has it just been neutral, right? So you knew how to test for this. Now, that's a fine story. But we need some data, right? What data? Where is my... So of course there's data out there. So this little table is going to show you amino acid changes. At sort of two different sites. The antigenic sites, which are the hemagglutinin and the neuraminase sites of the viruses, but we need not sort of worry about that right now. Just antigenic sites. And the non-antigenic sites, so amino acid changes. And then we're going to look at, on the left-hand side there, is basically surviving lineages and then extinct lineages. Because viruses is very, people who study these things are very fortunate because they have sort of lots of collections of viruses, ones that have stuck around for a long time, and ones that have sort of disappeared. Because we've been collecting viruses, really, we have a good data set from 1968 forward, because 1968 was a big giant pandemic, and we started really paying attention to it. There was of course the big one was 1908.
Right. Spanish flu.
Spanish flu. 20% of the world's population dies because of the flu. Mostly you're all ages, by the way.
It's unusual.
Yeah, that's what.
Ideal host.
So this is the number of amino acid changes detected at antigenic sites in surviving lineages versus extinct lineages, and non-antigenic sites in surviving lineages and extinct lineages. What can these data tell you?
Well, you know that the amino acid changes are more prevalent in the antigenic site for the survived viruses.
You agree? Amino acid changes in the antigenic sites are more prevalent in surviving lineages than
Extinct. Or
lineages that survived but did not change antigenic sites or other changes in sites. Right? Now, does changing something about the antigenic site guarantee your survival?
no.
No. But it might sort of hedge your bets on it a little bit, right?
Yeah.
But it seems like, and the data for the non-antigenic sites supports this idea. When you do this kind of test, they tend to be evolving at a neutral rate. The targets of selection, adaptive selection, seem to be the antigenic sites, and the data is now supporting that by doing these sort of McDonald-Kreitman sort of tests. Okay? So adaptive selection is occurring at the antigenic sites. There is definitely a difference in fitness among individuals that change antigenic sites. So there's, with respect to genotype, there's a difference in fitness. So you definitely have some adaptive or positive selection going on at those sites. Does that make sense? See? You knew how to do that. Right? So if you're asked the question, is the flu is it evolving by adaptive means or not, you'd be able to say something like, here's the information you need. That's what you need to sort of sort out as you get asked the question, what information do I need to answer this question? Now, in 1968, there was a pandemic, meaning a worldwide flu epidemic. Not as severe as the 1981, but definitely sort of a nasty one. And what they discovered is that the flu virus was related to swine. The way viruses are named is like H some number, N some number. I think this was H3N2. But
Sounds right.
I don't want to sort of promise you that, but it's not really important that you remember which strain it was. If you go to the CDC and work there, then it is important that you remember which strain it is. But apparently, this pandemic might have been related to, like, when they sequenced the swine, the virus that was sort of responsible for the pandemic. They discovered it was related to swine. How did they discover that? What tool in your toolkit of biology stuff would you use?
The receptors were specific.
Sequencing to do what with?
Viral DNA to see, because sometimes can
See what proteins are produced.
Well, what would you build with those sequences?
Proteins.
Vector.
Proteins.
No, I don't mean like recombinant, but I mean in a sort of data sense. If you had a bunch of sequences for viruses from cows, pigs, birds, humans, simian, whatever, what would you do with that? You would make a phylogenetic tree if you were a good biologist, wouldn't you? Because you want to know the relationship among them all. And so how do they figure out that virus had swine flu in it? Well, they built a phylogenetic tree, and guess where that virus sort of shows up? The swine virus shows up in the human lineage, and that's how they know. So it turns out, just to sort of make a long story short, that bird flu and human flu can sometimes sort of end up in swine together. And so they can mix and trade stuff with horizontal gene transfer. And when you have this sort of horizontal gene transfer, you get a very new strain of virus. And sometimes new strains hit populations really hard at first. And we'll talk about sort of virulence in a second. But generally, new strains of pathogens tend to be sort of more virulent, and then they sort of wane over time a bit. So, building phylogenetic trees, using McDonald-Kreitman tests and data about fitness and sequence to basically uncover and do the epidemiology of the flu. So, evolutionary biology has some use. And it is important to know So remember, adaptive selection seems to be going on at the antigenic sites, but not the other sites of host. So now you could have taken a guess at that, right? But that would have been called a hypothesis. Now we have data to support that idea, to say that the flu is adapting to the immune system. And we know exactly what sort of site that's happening at. Got it? Questions? Concerns? Cares? Nobody has any cares? You just want to go on break, don't you? Believe you me, so do I. But first, two more things.
Wait, so what were you? I do have a question. What were you saying about the neutral model then?
So the antigenic sites are evolving by adaptive selection,
Right.
right? Positive selection. The other sites in the virus genome are
Are-
not. They're evolving by neutral.
Is that because when there was an amino acid change in an antigenic site, more of them survived? Is that kind of what we're supposed to get from that?
Yeah. From this table, so there's a difference in fitness between individuals that change the antigenic site and individuals that do not. They have changes in other places.
Okay.
So more lineages survive that change stuff about the antigenic site. But again, just changing antigenic site doesn't guarantee your survival. Virulence. Now that we're talking about flus and stuff, let's talk about virulence. Why not? So this is the harm done by the pathogen to the host during infection. Virulence, generally speaking, harm done by the pathogen to the host. This ranges from lethal to no symptoms. So there's a wide diversity of virulence or the effects of pathogens on hosts. Lethal being something like Ebola, some of the more severe strains of cholera, things like that. No symptoms. Sometimes you can be carrying a cold around with not much symptoms. You can have herpes and not really have any symptoms. Yes, it's true. So, not always obvious, right? But as an evolutionary biologist, and when you put that hat on, one thing you should sort of ask is, why such diversity? Why such diversity in virulence?
Generally because the pathogenicity of a lethal strain it's more harmful to the host. So a good virus doesn't kill its host, right?
Good virus.
Or like a well-structured virus, I should say.
Actually, we'll get to that.
Okay.
I will offer you 3. The number shall be 3. No less than 3. 3 hypotheses about sort of virulence and how it works.
You have receptors for the pathogen or the symptoms, maybe caused from the virus or bacteria or pathogen. Is that what we need?
I don't know. So these are 3 hypotheses about virulence and sort of how virulence sort of occurs or why it may be or may not be so severe. First one is the coincidental hypothesis. As an example of the coincidental hypothesis, I will use Clostridium tetanus. Which causes tetanus. tetanus, the name. So, who is Clostridium tetanus, and where would you find him if you were looking for him?
On the ground.
In the soil. Its regular old habitat is in the soil or on rusty nails.
Yeah.
So here's the idea, here's how the coincidental hypothesis works. Basically, Clostridium tetanus is not seeking to invade your body. It did not cleverly put itself on a rusty nail waiting for you to walk along and step on it in your bare feet. Right? It had no intention of ever entering your body, ever. But it does live in soil and produces some pretty nasty toxins, neurotoxins, to us. But pretty nasty stuff because the soil environment is kind of a harsh environment. Pretty competitive as a bacteria. Got fungi trying to kill you, all kinds of stuff. So the infection or the virulence of tetanus to the host is really by just coincidence, right? There's lots of stuff that gets injected into your skin all the time, right? Just because you have a breach, you don't necessarily end up with tetanus all the time. But tetanus, if it ends up in your skin. So it's sort of a coincidence in some ways. Bad luck for us that tetanus causes, or Clostridium tetanus causes lockjaw and So do you understand? Coincidence. There was no intention of the pathogen to invade you as a host. So the virulence of that interaction is really just coincidence.
So why is it that they always suggest uncomfortable feasibility when you're a tetanus shot?
Because even though you've had a tetanus shot before, it doesn't hurt to sort of make sure.
It's just because it's the most worrisome, or like the most serious infection.
It's easier to get a tetanus shot because the vaccine is cheap and readily available than to sort of hope that you don't get tetanus. Right? So the next one is the short-sighted hypothesis. So we'll use the polio virus as an example of the short-sighted hypothesis. So where does polio live? The polio virus? In water supplies. On any given day, in the past, you could sort of drink water from the water supply and polio would sort of pass right through you. Just because you ingested the polio virus doesn't mean you were going to get polio. Polio sort of normally passes through the gastrointestinal tract and sort of leaves in the feces and goes on with its merry little life and doesn't necessarily infect you. But what happens in polio is the polio virus is going to cross the gastrointestinal lining and enter into the rest of the body cavity. So if you think about it for a second, your from your mouth to your anus is actually external, right? That's on the outside. We're just sort of a tube within a tube. As gross as that may sound, that's what we are. So once it crosses that lining, now it's got all this kind of habitat to exploit. And it prefers nervous tissue. But the problem is polio virus then will never leave the host. It doesn't have an exit. Right? Its normal way of getting out is through the feces. Once it invades the nervous tissue, it doesn't get out.
Right.
I mean, that's bad for the host, right? Usually bad for the host, but it's also not so great for the polio virus, right? Because now that lineage of polio virus is just hit a dead end. Not to feel bad for it or anything, but but you see the idea, like it's now sort of not going through its normal life cycle and is not going to be passed on to the next host. It's stuck in the host. So that's the short-sighted hypothesis. And the last one is about trade-offs. Is that boring?
So I have a question. So what's really the difference? How is that not coincidental? If it doesn't intend to go through the gastrointestinal lining, how is it not coincidental?
Yeah,
What's the take on?
they are sort of related in a way. They're just, this is just, there are a couple different ideas out there, and these examples are put up for them. So this is also sort of coincidental in a way, yes. So there's a bit of crossover. The trade-off hypothesis. Here we have natural selection should favor a balance between virulence and transmission. So just like any other good old trade-off, there's going to be some kind of balance, some kind of optimum. Just like in economics, just like in ecology, there's going to be some kind of optimum with a trade-off. So natural selection is going to favor some balance between making your host a little bit sick and getting out of your host for transmission. The idea is that the probability of transmission goes up as virulence goes up, but at some crucial point, it goes back down. So that the optimum is going to be right sort of at the peak, where you make your host just sick enough to sort of, so that you can get in there, take down their system, use it for a while, and then get out. And move on to the next host. Yeah? See that? Often, new varieties, or I guess strains is a better or more appropriate, when they enter new populations, when new viruses, new pathogens sort of enter populations, or like I said, swine is something like that. When you get something that hits a population new, it tends to be the virulence is sort of a little too strong, right? It's just a general trend. It's not going to happen all the time because this is biology, but you do tend to see when some new stuff tends to sort of fall over here, and then of course natural selection is going to push it back. Because basically it's going to make the host too sick, and it's going to make the host so sick that the host is going to die before the thing can get out. So the strains that are too virulent are selected against because they never make it out of the host. The host dies before they can get out. Right? So the whole virus thing is to go in, do your thing, replicate, and get out and move on to the next host. But if your host dies while you're in it, you don't get out. Right? So if you're too virulent, then that's a bad thing. And so the probability of transmission goes down. So selection is going to push it back. One more thing here. Is that so there's another point, so we'll call this A. There also tends to be a difference in virulence between pathogens that have sort of different life cycles. Some pathogens, like the rhinovirus, the cold, are directly transmitted, right? If you have a cold and you go spit on your friend, and, spit in their face, then they're going to get a cold, right? Or they borrow your pencil after you've been snotting all over it. Right? So that's a direct transmission. But other things like malaria have an intermediate host, right? There's a mosquito involved, an intermediate host. Now, so the inflection point on that graph is different usually between these two. Again, don't quote me on it that everything is like this, but so with an intermediate host, we use malaria as an example. With a direct, we'll use the rhinovirus, common cold. Where do you think letter A, is that direct transmission or intermediate host virulence sort of optimum?
Direct.
Direct.
Direct? How many votes for direct?
What? Yeah.
So intermediate host and direct transmission, which one's A and which one's B?
I think A is direct transmission.
Low.
See, this is where those little clickers would be great, right? It's worth the $50 or whatever you gotta pay for that, right?
$10 for that.
$10 for that, but then you gotta keep it up, right? Yeah,
Huh?
I'll fight as long as I can, y'all.
Maybe say
They have their value, sure, but I'm not sure what that is. Actually, the only value I've seen is that you can take your exams on it, and then, like, basically I don't have to grade crap. It's all graded for me, and like, oh, you're. What's the name of the clicker?
Virulence. That's their peaks, right?
Yeah, so A would have low virulence, so the inflection point for A is lower here.
But higher transmission at a lower.
Lower virulence, right.
That's what needs.
So it's two, so what, so intermediate host, A or B?
B.
A.
All right, so it's kind of mixed, this would be B. So in the case of malaria, and again, it's not always true, but in the case of malaria though, well, at least direct transmission, let's not do malaria, let's do cold. If you're laid out by a cold and you're not moving around, then the probability of transmission goes down for that cold because it in some ways wants you out there walking around and sneezing on people.
Right.
Because then its transmission is best, right? So it doesn't want us. You might feel a little bad, but like, how many people stay home like for just a cold, right?
No,
I
it's
do.
like real bad.
I think I can't get it. Any excuse I can find, good enough. But seriously, you can usually get up and go around and spit on your friend, as I said. But in the case of the intermediate host, the malaria would rather you just sort of be laying there so other mosquitoes can come and fight you.
Right.
So it's to the advantage of the. So the inflection point is going to vary on the life histories and then the mode of transmission. That's the point. Do you understand how that works?
Yep.
Good. So then last but not least, right?
HIV considered another intermediate type of thing?
No,
It's not?
no, that's directly true.
Yeah, because HIV is
Well-
infected in a person, then another person infects them. Intermediate would be like malaria or bubonic, where something bites them and takes the blood.
Well, that's kind of why I guess I was thinking like blood or bodily fluid transmission. I wasn't seeing the relationship.
I mean intermediate, like another organism.
Okay, I got you.
Like what?
Like rabies. But rabies can also be direct transmission. Just depends on how you look at it.
Virus.
The whole thing's going to fall apart if you start analyzing.
I know.
Just get enough data to prove your point and move on. Never sort of get a bigger data set. Bigger data sets are overrated. I didn't say that. I never said that. Erase that.
I'll have video editing.
And one last thing, fever. Is fever adaptive?
No. Fever is a secondary symptom of infection.
Like, you get a fever when you get the flu or something, right? Is that good for you?
No.
Or is it good for the bloody invader?
It's good for you, but it's bad for the virus.
It's a defense.
Generally. Right.
I mean, it can't.
Okay, so we have two choices.
But it depends though, because certain viruses prefer.
Good for you. Good for invader. And does anybody know the third choice?
Good for both. Bad for both. Bad for both.
Yeah. Well, I'm gonna just do good.
Or both A and C.
Good for none. Okay.
All right, so now we have 4 viable hypotheses about is fever, like what's fever good for anyway? Is it good for you? Is it good for them? Or the pathogen? It's good for both of you?
It can be.
Or is it actually good for none of it? And it just happens, right? Because it's possible that you get a fever and the pathogen hates it and you hate it, and then nobody gets any benefit from it, but your body does it anyway. Like all the snot you produce from a cold, it's like your body overreacting just a tad, usually. I'm sure you need to flush it out, but that much? So you see there's a number of different options here. If it's good for you, maybe it's helping you sort of fight the pathogen somehow. If it's good for the pathogen, well, you know bacteria and such things, they have higher replication rates at higher temperatures, right?
Right, so
So they can reproduce faster if they can elevate your body temperature just a bit.
Right.
So that's good for them. It could be good for both. It could be good for none. What do we need?
Data.
Data, right.
Here.
Let's of course start with iguanas. Why not? Of course. An ectothermic poikilotherm. Right? Ectothermic. Right? It basks. And poikilothermic meaning variable temperature. So you can have ectothermic homeotherms and endothermic homeotherms. And poikilotherms. You can have them all mixed up. So an ectothermic homeotherm would just be something that regulates its body temperature according to the outside, but the outside never really changes. Like someone who lives in the deep ocean or whatever.
Right.
Stable body temperature is homeothermic. Variable body temperature is poikilothermic. Ectothermic meaning external source of heat. Endothermic meaning heat generated on the inside. But these are ectotherms, iguanas. So what some people did with some of their iguanas, what some scientists did, is they designed a little study. And of course the study had controls and all that stuff, so it was well done. But they basically infected some iguanas with an iguana pathogen, a bacteria. And in some cases they didn't let the iguanas bask at all. In some cases they only let them bask a certain amount of time. In other cases they let the iguana bask all it wanted. And, you know, so this is a more elaborate experiment, but the iguanas that were able to bask all they wanted would elevate their body temperature 2 degrees centigrade on average above what they sort of normally would do basking. So they knew their normal habits of basking. The iguana would bask for a certain amount of time until its body temperature reached a certain temperature. And then it would say, oh, all done, and it would go about its business, right? Well, when it was infected with a bacterium, it would wait a little longer and elevate its body temperature 2 degrees Celsius above its normal temperature. When they were able to do this, when they were allowed to do this, the group that was able to do that had an increased survival rate post-infection and was able to fight off the bacterium or the pathogen better. We call it behavioral fever. Or at least that's what it's been dubbed. When the iguana was infected with a bacterium, it intentionally gave itself fever by basking just a little longer. And by doing that, it was able to fight the pathogen off better, have higher survival rates, things like that. Is fever adaptive? Is it good for the invader? Is it good for the host?
Good for the host for the most part, but it can be good for viruses because certain viruses reproduce at higher temperatures.
So we're still undecided, but this is definitely good for the host at least.
Right, in this case.
So we have one vote for it. So this is definitely sort of, yeah, here, and it's hard to tell there, maybe. But we definitely have one. The problem is these are iguana and not endotherms.
Right.
Which of course is going to be, and us being endotherms, we'd be more interested in that. So, I'm certain there's been more work done than this, but I'm not familiar with any of it, so I'm talking about the rat work that I do know something about. So the problem with endotherms is this. You can't increase your body temperature without necessarily increasing your metabolic rate. They sort of go hand in hand, right? So body temperature and metabolic rates are linked. Same with the guano. But what this rat study was trying to sort out is fever, what about fever is helping us? Is it the increase in metabolic rate, or is it an increase in the body core temperature that's really sort of useful, if it's useful at all? They did some clever things where they were able to raise the metabolic rate of the rats without raising the core body temperature. So we did some clever little things. And in the rats that were able to increase the metabolic rate, the treatment that stood out as the sort of most effective at fighting off the pathogen was when the metabolic rate was increased, but not the body temperature. I don't know how we could do that. Drink a lot of coffee or something when you get a cold. Those energy drink, whatever things. Shouldn't drink too many of those.
What?
So I always thought that was sort of an interesting little thing. It seems like the increase in metabolic rate is a benefit to the host, but the increase in temperature is actually a benefit to them. So here we have sort of a vote for fever is good for you because it increases metabolic rate, good for the invader, so it's sort of good for both. So in some ways it seems to favor both. And then it, I guess, becomes a race. Can you kill it before it kills you?
Right.
So, what do you think about taking medications that are supposed to reduce the fever?
Oh, let's talk about that.
You know, I mean,
How did-
that's,
How'd you get that it was good for invader from that?
Huh? Good for who?
For the invader from that.
Oh, because increased temperature didn't necessarily make survivorship go up. So the only factor that came out as increasing survivorship was not increased temperature, but increased metabolic rate. So when you increase temperature too, survivorship was no different than individuals that were not left. So actually, let's talk about just some data real quick. So this is old data, old news. Older than you. Some of it. But again, I think it's sort of fun. And I think it's a good exercise in what data can and what it can't study. So here's a study from a long time ago by Graham and that famous scientist et al. So on the y-axis is going to be the titer, or the sort of how much anti-rhinovirus antibody was produced by the organism, by the people.
28 days later.
Right? So that's just a measure of sort of immune response. How much antibody has been produced to the antigen?
I think you're the only one that caught that.
The individuals were sort of injected, if you will, or inoculated
Right.
with the rhinovirus, the common cold, and then given different fever medications, and then they tracked a number of things, including the anti-rhinovirus antibodies. They were given, some individuals were given the placebo. So there's a placebo, ibuprofen, acetaminophen, and aspirin. So those are the different treatments. So those are all, besides the placebo, which was the sugar pill, those are all anti-fever medications, or something you might take should you have a fever. So, here they are, the data, as they sort out. The labels on the other side are in no particular order. What I'd like you to do is sort out what order they were in. So, where do you think the placebo is?
Bottom.
Bottom. Okay, so this might be P. And then aspirin,
The dashes.
ibuprofen,
Top.
let's see.
Might have those backwards, the last two. But yeah, I think so too.
All right, you got one right. So aspirin, you got right aspirin.
Let's go for placebo at the top.
So you go to the top then.
Yeah.
Okay?
Okay, sorry.
So
Ibuprofen at the bottom.
What can be said of these data? What do the anti-fever medications do to the titer of the anti-rhinovirus antibodies? The individuals, so those are all sort of significantly different from each other, by the way, statistically speaking. So they all produce a different level of titer. The placebo produced the greatest immune response. The greatest amount of antibody was produced when the individuals did not take any anti-inflammatory or anti-fever medications. Right? And acetaminophen was definitely sort of the one that, what should I say, suppressed the immune system the most?
yeah.
Yeah? So is it fair to say from these data that anti-fever medications interfere with the immune response
Based on this.
Yes.
for rhinoviruses?
And rats.
You have to be specific. You cannot take these data and extend it to everything. And you also have to be aware of they suppress the immune response with respect to antibody production for anti-rhinovirus. Does that mean taking fever medications is bad?
No.
No.
Right up there.
Right, if your fever is very high, you gotta bring it down. So, but there's definitely, the data's the data, like, there was definitely some interference going on. Is it a terrible bad thing? I don't know, but also I would say that most people don't really take anti-fever medications when you have a cold
Right.
much, maybe when it first hits you or something, but.
Well, I mean, if you take Dayquil, any of that stuff.
Yeah, so that's not
all
included
that
in here, right? But that's got, some of that's got
Yeah, it's got
benefit.
all that in there.
It has
Always.
dextromorphin and a bunch of other things too.
Medicine whatsoever.
So, this is not, you have to be careful with the data, right? It can say that they interfered with the immune response, but that's all. You can't sort of make big conclusions after that, so be careful about it. And this is just for the rhinovirus, and this is a published study in 1990. I'm not quite sure what's been done. I've never, they don't mention this on the commercials. You don't see that, or you don't hear the guy with the voice going, "Could cause diarrhea, blah, 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 blah. That affect the immune response," that's not in there. Which probably should be, but, anyway, I found that kind of fun. Now, is fever adaptive? Maybe. Suppressing fever, you have to in some cases. But it's nice to know that these things actually, they can affect your immune response. And, how severe is that really good? There's some more data in that paper that suggests that when you take the acetaminophen and things like that your cold will last longer actually, because you're suppressing the immune response. So that makes sense, right? It takes longer to build the immune response, so you end up hanging on to the cold a little longer. Some of the symptoms there. Any questions on that? Good, clean fun, right?
Yep.
Have a good break. Make sure you get some rest. On Tuesday, there is a study guide up. If you have questions, ask.